Kombucha is a very special type of drink that has gained vast popularity in the 21st century in Western society as a result of its unique taste and claimed health benefits. Word of kombucha's health benefits only reached Europe via Russia in the 20th century, but now it's being developed into a commercial beverage that's being sold worldwide, and you can find it almost anywhere, including your local supermarket. So what exactly makes kombucha kombucha?
So we've come to Bennett's Kombucha in Ilminster to find out. So this is where they make the kombucha, and here we've got a few crates of flavoured bottles which are just about to be sent out. Let's go meet our client, Neil. Hello, Neil.
Hello!
What are you doing in there?
This is the Scobie Hotel, and this is where we do all the fermentation of Bennett's Kombucha.
Yeah, right. Why is gut health so important, Neil?
Because as a nation, we generally have poor gut health. We've suffered from COVID, and tragically more people have died because of that poor gut health. We eat too much processed and ultra processed foods, but also increasingly we've got a population that's overweight and obese, and I think that has a direct link back to their guts and back to what people's microbiome is made up of in terms of beneficial bacteria. So gut health has a huge role to play in both our national well-being but also our individual well-being.
Could you then tell us why kombucha is important in combating that poor gut health?
So we got into the whole kombucha business because Tamara Bennett, who's a registered nutritionist, understood the gut health and the importance of beneficial bacteria, and that our three major health systems, so metabolic, mental and our immune health systems, are all reliant on that gut and that microbiome, which needs trillions, if not Googles, of beneficial bacteria. And therefore kombucha is one way we can invest and get that bacteria into the gut, which is so fundamental to our well-being and to our fundamental living.
At its core, kombucha is a fermented tea. Preparation begins by steeping the tea leaves in boiling water for 10 minutes. The most common tea used is black tea, however white, green and oolong teas can also be used too. The tea leaves are then removed and table sugar or sucrose is added. Once this mixture has cooled to room temperature, a small volume of previously prepared kombucha is then added, followed by a symbiotic culture of bacteria and yeast, or SCOBY, which is in the form of a solid pellicle. The mixture is then left to incubate for 7 to 10 days. So, just as a quick recap, kombucha is a fermented tea originating from East Asia. Currently, our nation's gut health is extremely poor as a result of eating highly processed foods. Research has shown that the bacteria in kombucha can improve gut health, helping to increase metabolic, immune and mental well-being.
After visiting Neil at Bennett's Kombucha, he left us with the main objective to maximise the health benefits of his kombucha. To do this, we are looking at two main characteristics. The first one is to maximise the amount of polyphenols that are in the kombucha, and the second one is to characterise the bacteria that are present in his current kombucha and maybe look at what factors will get the most beneficial bacteria out of the kombucha. Polyphenols are a type of antioxidant which reduce oxidative stress. Oxidative stress is this excessive synthesis of reactive oxygen species, also known as free radicals. These molecules can travel through cellular structures, causing damage. They cause cell death, tissue damage, which can then lead to Parkinson's, Alzheimer's, and even cancer. There are many factors that can influence these polyphenol and the bacterial content, but we are looking at three main ones. So the first one we're looking at is the type of tea that is used as the base of the kombucha. Neil and Bennett's Kombucha, they mainly use white tea, organic white tea, but most of literature is dominated by studies on black tea. The polyphenol content between the two varies due to their manufacturing methods. Organic white tea is said to contain more polyphenols than black tea. So we want to see what would happen if we compare the kombucha made with a white tea base to the kombucha made with a black tea base. So the second factor we're going to look at is the age of SCOBY and if that affects the final kombucha product. We're going to look at a younger SCOBY called SCOBY1 and then a second SCOBY which is older by around a month, and see if that affects the final product of the kombucha. A previous paper has seen that older SCOBYs have a high proportion of lactic acid bacteria, which are more beneficial for your gut health. So the final factor we're going to look at is something that has not been done in literature before. What we're going to do is add blackberries to the initial stage of kombucha and let them ferment the whole time with the SCOBY. This is important because blackberries are one of the richest sources of polyphenols. So adding those initially will hopefully increase the amount of polyphenol content of the final kombucha product.
Neil wants us to optimise his recipe by maximising the amount of polyphenols and probiotics in his kombucha. To do so, we will investigate 3 variables in the process to see which will give the best outcome. These are SCOBY age, tea type, and addition of blackberries.
Our experimental design involved 8 batches of kombucha, 4 using black tea and 4 using white tea. For each tea, we divided the batches using 2 different SCOBY samples, then finally adding blackberries as another variable to 2 of the kombucha batches containing a different SCOBY. In preparation for the kombucha making process, 8 10-litre tubs were cleaned using boiling water to contain the starting fluid and SCOBYs. For each tub, 2 litres of boiling water and 20 tea bags were added, 4 tubs containing white tea and 4 tubs containing black tea, and allowed to steep for 20 minutes. After 20 minutes, the tea bags were removed and 250 grams of white sugar was stirred into each tub until dissolved, followed by adding 3 litres of cold water. For the 4 batches containing blackberries, 1,000 grams of blackberries were washed and blended and split equally between the 4 tubs and mixed well. Each tub contained 224 grams of blended blackberries. And the tea was left to incubate and ferment.
During the incubation period, the yeast in the SCOBY use an enzyme called invertase to hydrolyze the sucrose into fructose and glucose. These metabolites are then fermented into alcohols and carbon dioxide, which gives kombucha its natural fizz. The bacterial population in the SCOBY is dominated by acetic acid bacteria belonging to the genera Kombucha2022, Glucosinobacter and Glucosinacetobacter. These organisms further oxidize the alcohol content present into organic acids, which makes kombucha slightly tart, but also importantly inhibits the growth of any pathogenic species. The end result is a product teeming with supplementary compounds that have profound impacts on human health.
Each week we revisited the kombucha batches, analysed and documented their appearance, smell, texture, thickness and growth. To measure the pH and sugar content, a cupful of liquid was collected from each batch at the edge of the tub to avoid disturbing or damaging the SCOBY. After all tests were conducted, triplicate samples were collected from each of the leftover kombucha glasses.
We have now made all 8 of our kombucha cultures and are going to leave them for 3 weeks to incubate.
The FRAP assay protocol was used to measure the antioxidant capacity of the kombucha samples. First, we had to calibrate the spectrophotometer. We then had to plot a calibration curve. The calibration curve was established by adding 1 milliliter of FRAP reagent to 6 cuvettes with 25 microliter standards of ferric sulfate at different concentrations. The calibration curve was then plotted using the known concentrations and the corresponding absorption values. We then conducted the FRAP assay on our samples. To a centrifuge tube, 25 microliters of the diluted kombucha sample and 1 milliliter of FRAP reagent were added and then vortexed. Each cuvette was then inserted into the spectrophotometer and the percentage absorbance measured at 593 nanometers. From the calibration curve, these recorded absorbance values were used to read off the antioxidant concentration for each kombucha sample. For the polyphenol assay, we followed a very similar protocol but with gallic acid instead of FRAP solution. This gave us the relative absorption, which we then determined the concentration from using a calibration curve once again.
In order to identify the bacterial genera present in our kombucha cultures, we carried out sequencing analysis of the 16S rRNA gene. This gene is present in nearly all archaea and bacteria in 1500 base pairs and encodes for a small subunit of an important ribosome. Although it is highly conserved, this gene sequence contains 9 so-called variable regions where mutations will not disrupt the functionality of the ribosome. These areas can be used as a barcode to identify and distinguish between different taxa of bacteria. We performed this sequencing on samples of the 2 scobies prior to fermentation, as well as the resulting scobies and kombucha liquid post-fermentation. The procedure for sequencing began with extraction of DNA from within the cells, which involved first mechanically breaking up our samples. Then our samples underwent several incubations and centrifugations with digestive enzymes to cleave open the cells and release the genetic material. Ethanol was then added to elute the DNA from solution. We then took our extracts to the Exeter sequencing service, where Illumina sequencing was used to determine microbial contents of our SCOBY and kombucha. To recap, we measured the total polyphenol content and antioxidant capacity of our samples every week for the 3 weeks of fermentation. This was done using 2 standard assays. We were then able to extract DNA from our samples and give it to the Exeter sequencing service so that the bacterial species could be identified.
So now after 10 weeks of hard work, we are on to the exciting part, the results. Right, so we're going to start with the least exciting result. This is comparing the white and the black tea. Although we predicted white tea to have a higher phenolic content with the final kombucha products, we actually saw that our results were kind of inconclusive. This has an easy explanation though, because both the FRAP and the total polyphenol content, because they use that spectrophotometer, the absorbance is based on the colour of the samples. Comparing the white and the black tea, the samples are already different colours to begin with, so the absorbance is not actually indicative of the total polyphenol content. The way we improve this in the future is using the more expensive HPLC method to actually characterise all the phenols present in our final kombucha. So now we're going to look at the difference between the younger SCOBY 1 and the older SCOBY 2. This doesn't involve that colour difference issue, so these are quite significant results. You can see that for both the total polyphenol content and the FRAP, the kombuchas created with that older SCOBY actually have a higher total polyphenol content after the 2 weeks of fermentation. This is because, as I said before, the older SCOBYs have a higher proportion of lactic acid bacteria, which break down those large polyphenol chains into phenols that you can absorb easier into your gut. So finally, we're going to look at the addition of blackberries into our kombucha. We found that the total polyphenol content and the antioxidant capacity of the liquid does increase when we add those blackberries. This is rather derivative because obviously those blackberries have additional polyphenols, and when we add them, the total amount does increase. But the important finding is that those additional ones stay throughout all the stages of fermentation, so that the end results are still significantly larger than those without. So let's move on to what, in my opinion, is some really intriguing and promising sequencing results. Right, so using just that 16S sequencing, we were able to see that both of the initial SCOBYs are completely dominated by that Kombucha genus. This is a group of bacteria that's responsible for making the cellulose that forms that solid SCOBY pedicle that you see at the top of all the kombucha. So let's compare that younger SCOBY 1 to that older SCOBY 2. You can see that the relative abundance of the different genre of bacteria is almost identical. And you can see that there actually aren't any lactic acid bacteria identified at all in either of them. So this is mainly down to the database that we use. We use quite a relatively small database. So in the future, if we use a longer process that uses a much larger database, we could correctly label almost all of the bacteria present and reduce that uncertainty. So these are the 8 sequencing results after that fermentation. The really cool part is once you compare the black tea to the white tea. You can see that for the white tea on the left, they have 2 new genera of bacteria. This is a Meyermeyer and the gluconobacteria. These are both acetic acid bacteria that convert that ethanol into short-chain organic acids that are really good for your gut health. So from that, we can hypothesize that white tea might promote the growth of acetic acid bacteria more than black tea. The exact mechanisms of this have not been researched yet, so that's a really exciting potential future research. If we compare those 2 initial SCOBYs before fermentation to those 8 final SCOBYs after 3 weeks of fermentation, we can see that the sequences found are significantly different. This proves that the fermentation process does change the bacterial composition of those SCOBYs. So that means our overall winner is the kombucha made with a white tea base, an older SCOBY, and having blackberries added into it. But what does it taste like? Well, a taste panel of 20 participants has shown that white tea with SCOBY 1 and 2 are significantly favoured over the rest, with those both being the top answers for samples people would drink again. Unfortunately, the white tea with SCOBY 2 and blackberries on average ranks last out of all of the 8 samples, as the blackberries create quite a sour final taste. So, perhaps the exploration of other polyphenol-rich sources with a more mellow taste, or potentially adding the blackberries at a later stage of fermentation would produce kombucha that is great for your health and pleasant to drink.